Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Practical AI Adding Insights to the Patient Journey with Digital Phenotyping. I'm Sydney Otamanchak from the events team at Scientist.com, and I'm happy to be your host for today. In this webinar, Dr. Joseph Zabinski, the Vice President of Commercial Strategy and AI at OM1, will discuss the digital phenotyping process and several concrete applications to demonstrate how AI can add value to analyses of the patient journey across the life sciences spectrum. I'd like to thank the sponsor of today's event, OM1, for making it all possible. And now, without any further delay, I'm pleased to welcome our speaker, Joseph, to lead us into the presentation.
Great. Thank you for that kind introduction, Sydney, and thank you everybody watching for taking some time out of your day today. Hopefully, this will be a good use of an hour of your time as we try to get practical with some of what AI can do specifically in helping to enhance our view into patient journeys. So before we go into any of the AI stuff, I figured I'd just briefly define what I mean by the patient journey. And of course, this means different things to different people, but everyone has some intuitive grasp of what a patient journey is. And I think it's fine and appropriate to take a pretty expansive view of what we mean by the patient journey. This is a description of the patient's experience from wherever that experience starts, for example, with their initial symptoms, through their diagnosis, through their treatment, and through long-term condition management. Of course, those few words describe a journey. It is not always that simple for individual patients, particularly in many complex conditions, but that's what we mean by a journey. The notion of mapping this patient journey is really intended to allow us to better understand what's going on with patients, what they're experiencing, and ultimately the needs that they have, including those that are unmet in current practice as they move from symptoms through diagnosis, treatment, long-term management, and sometimes kind of recursively loop through that pathway. And we'll talk about what some of those unmet needs are and how digital phenotyping can help later on during today's webinar. The notion here as well, and this is something not unique to this presentation, but that I think we've been observing in the industry for several years, is that with the broader and richer data that we now have that have really been coming online with the advent of new technologies, new data availability, as well as some newer analytic tools like AI, we have greater and greater ability to gain insight into the patient journey to understand and close critical gaps. So, a lot of times we try to do the best we can with the data and tools available to understand patients' journeys. But at the same time, we're more and more aware of what's missing from those journeys. So the intent here is to use these newer data sources, these newer tools to identify and then close gaps that exist. What are some of those gaps? How are patient journeys incomplete today? Of course, there's lots of ways that we could tackle this problem, but just to highlight a few of those, I think it often comes down to this notion that patient journeys are usually constructed from limited data samples, and because of that, they miss real-world insights into what's actually going on with patients. Traditionally, a lot of patient journey construction, particularly in the life science industry, has a reliance on clinician interviews in relatively small data sets that may not be fully representative. Of course, it makes sense to ask clinicians what's going on with patients in a disease that they're familiar with. They're the experts. The problem here is that there's only so many physicians that we can interview. And when we're looking at small data sets, there's only so many sort of anecdotes that we can analyze. These methods do provide a valuable starting point, but they can miss nuance. And in particular, they can miss heterogeneity in the patient population, right? People don't think about those folks who are not immediately in front of them in their mind when they call to mind a patient progressing along a particular journey. And when amplified, that sort of missingness of individual experience can translate into gaps in the journey that we can see as we try to understand what happens to the whole body of patients, not just those that most immediately spring to mind. A more subtle kind of concern or at least level of incompleteness with current journey analyses has to do with how those traditional methods of constructing journeys inform how the journeys themselves manifest. And in particular, this notion that journey mapping tends to be based on sort of linear and sequential logic. And I'm guilty of this too. I mean, even on the previous slide, I sort of laid out what is a journey. A journey is symptoms, diagnosis, treatment, long-term management. Very linear process, even with some verbal reference to recursion and looping. There's this tendency that we have when we try to lay down kind of what one of these things actually looks like to want to have things follow in a natural sequence, A to B to C to D. And that's certainly, again, something that experts prefer to do because they've studied these diseases, they've studied patient progressions, and they understand that each event leads to another, even though they also recognize that there's real world variation, real world heterogeneity. The challenge remains, though, that when we sort of over rely on a tidy narrative of what's going on with patient journeys, we can miss a lot of real world behavior, particularly in subgroups of patients who may not follow that same linear logic. Perhaps they have some symptoms and they get diagnosed, but it turns out they have a comorbid related condition that hasn't been diagnosed. They get a partially effective treatment, but they still have some symptoms. So they go back to the beginning of the process, and this linearity can be a bit of a trap unless we're careful about it. So it's something that we watch out for carefully. And then finally, this notion of missing data and the kinds of challenges that can introduce into patient journey understanding. I mean, of course, many folks have spoken at length, and the audience is probably familiar with challenges of missingness in data, as well as bias that can arise from missingness in data. But just briefly, there are those data gaps that we're aware of when we look at a data set. It can be obvious if, for example, we just haven't observed a patient's sex or their weight or something like that. We know that these are things that exist, we just haven't observed them. The more insidious form of missingness is when we're not even aware that data are missing, right? When we summon a cohort of patients who are diagnosed with a particular condition, and we don't know that, in fact, there are meaningful other groups of patients who have the same condition, but because they're not accessing care, or because their care is being provided, but their data are not making it to our analysis, we're just unaware of them completely. These are sort of the gaps where information is hiding. It's present out there in the world, but we can't see it. And that is one of these vectors by which bias can come into our understanding of patients, into their journeys and how they're treated, and ultimately translate into issues with those patients accessing care in the future. So we have to be very careful about this notion of missingness. Now, we understand that there's tremendous richness available in patient longitudinal data, right? Whenever we are looking at a patient's data, even with the caveats I've mentioned around missingness and so on, if we take relatively raw data from as many sources and as many different ways of accessing that patient's experience as we can, and we put it together sequentially, there's quite a lot of insight that can be gathered about some of these questions that I've already started to mention, right? When did a patient begin experiencing something that could be associated later with a disease? How did they fare once they started a particular treatment? Can we observe things about whether that treatment worked, or why they switched off of that medication, for example? These are all things that we all classically understand go on with longitudinal healthcare data. The challenge historically has been, how do we condense and collect signaling information from across these long clinical narratives that we have, structured or unstructured, in the data, and recognize subtle similarities and differences across cohorts of thousands, tens of thousands, millions of patients that we can use to operationalize particular insights about a group of interest. That's really been a challenge because we can often see these insights when we read a particular patient's data or narrative directly. But of course, we can't read everybody's narrative using just our own processing. We need technology to help us identify subtle signals that patients with common characteristics share, that differentiate them from others, so that we can say these are the things that happen, both before and around and after the event of interest that we care about, that really help us flesh out this patient's journey. And that, in a nutshell, is the kind of problem that we solve using digital phenotyping. Digital phenotyping is an application of several different artificial intelligence and machine learning technologies that really, at its core, attempts to solve the problem I just outlined: the identification of common patterns in long scale patient, longitudinal patient health histories that are associated with things we care about. Those could be diagnoses, symptoms, treatment patterns, whatever it may be. Associate those patterns with those characteristics, and that can help us study and fill in those patients' journeys relative to others, and answer all sorts of questions that are left unanswered by the traditional methods we might have. At OM1, we've developed this phenome technology that does digital phenotyping. And in each case, we ask Phenome to do essentially the same thing under the hood. We say, here's a group of patients that we want to understand better. Let's say a group of patients diagnosed with a certain disease. Let's collect some of those in the available data that we have. Let's use this digital phenotyping tool to say, what do those patients share in their health histories prior to reaching the diagnosis we care about? that they have in common and that differentiate them from others. And then once we understand that set of signals or features in their history, we can look for other patients, for example, who match that pattern. I think of these things as fingerprints, the fingerprints being made up of these different signals that are isolated. And we can compare other patients' data to those fingerprints and say, is this someone who also looks like they're experiencing a similar journey? And can we say where that journey is more likely to go proactively, even before that journey has fully manifested for the new individual? Doing this lets us move much more fluently and fluidly back and forth in terms of patient journeys to say we can tie information from 3 years ago to what's going on with the patient today to what's most likely to happen to them a year or two in the future, understanding the complexity of their interaction with the healthcare system and treatment and so on. This digital phenotyping approach to understanding patient journeys has proven quite powerful in a number of different application areas, and I'll give 3 different case studies to kind of demonstrate this more fully in a couple minutes. Um, Before I go into those details, though, I just want to emphasize what we're really doing here in any case is looking, as I said, at a common set of patients, a set of patients, usually in the journey context, who have a common diagnosis or a common treatment pathway or some sort of risk that they manifest and that they share as a group, and saying, how do we most fully understand and capture the events in the past, present, and future that help us understand from a research and treatment perspective what's most useful to help those patients. You'll hear me reference OM1 Orion. This is our technology built on top of digital phenotyping that specifically applies it to answer questions of relevance to patient journey analyses. And just to give a bit more clarity on what I mean by these digital phenotypes, I referenced that they come from patients' longitudinal real-world data of health history records. But in terms of the little atomic pieces of evidence that live inside of them, they're kind of the things that are on the screen here. Of course, they're complicated mathematical objects, but at a descriptive level, it's fair to say a phenotype associated with a patient will often tell us something about that patient's prior level of disease activity, evidence of progressive disease, perhaps differences there where we can see information that a patient is moving faster or slower in disease progression than others, prior treatment history, including, what has worked and what hasn't worked, information about their physician, sometimes seeing patterns that are common among patients all treated by the same physician can be useful information to inform our understanding of a patient journey, comorbid burden, mental health effects, and lots of other things. All of these pieces of information go into a phenotypic profile associated with some characteristic of interest. And then let us say prospectively, here are patients that are similar to that profile. What does that tell us about their experience? How can that help us fill in gaps in our journey understanding of this disease? And finally, the kinds of questions that we answer using digital phenotyping as a process with respect to relevance in the patient journey are listed thematically here. And most of the time, the greatest relevant ones with respect to journey analyses are living at the top of this slide, right? I've mentioned diagnostic cases several times. Probably the most frequent question I get about digital phenotyping and patient journeys is, can you help me understand undiagnosed or misdiagnosed groups of patients who I should be thinking about as I think about the disease that I care about from a medical perspective, from a commercial perspective, whatever it is, but that I can't see for whatever reason using the data access and the tools that I have at hand. Can you help me, put the finger on those patients who are undiagnosed or misdiagnosed with my disease of interest? Treatment insights is the second most popular category that we can apply this digital phenotyping to. And here, the notion is really we want to understand when a patient is thinking about a treatment, everything that we can leading up to that decision, and then its consequences as that goes forward in the patient's journey. So, for example, we can observe classically, that a set of patients initiated a medication. And perhaps we can get some insight from structured data into why that medication was initiated. But we might want to say, of those patients who persisted on a medication 6 months or a year after initiation, why were they still persisting? Or, conversely, those who dropped off, why did they drop off? Was it an issue of coverage? Was it an issue of side effects? Was it the medication simply not effective? Did it resolve their symptoms to the point that they didn't feel the need for it? All of these are the kinds of gaps or areas of uncertainty that we try to fill in using digital phenotyping by saying, let's drill down on patients who display each of those different properties I mentioned, and then figure out phenotypically, is there a pattern we can see that's associated, for example, with side effects or with disease resolution, that we can pinpoint more accurately to say this is the path that patient A took versus B versus C. We also, typically answer questions around risk insight using this same technology. And this is, most easily described when we care about patient journeys with respect to catastrophic events, right? It is useful to be able to say which patients are most likely to experience a severe event like a heart attack or a stroke prospectively. Also, what was unique about patients in a disease population we're studying that experienced such an adverse event relative to all their other cohort mates who didn't experience that adverse event? Is there something phenotypically distinct about those individuals using the kind of data patterning that I've described? The risk insights can also answer questions we might care about with respect to things like risk of disease progression to a different condition or to a more advanced form of their disease, or the pace at which that occurs. Frequently we'll get asked to say, is there something phenotypically distinct about rapid progressors, right? From moment of diagnosis, some small group of patients seems to break off and progress much more quickly than everybody else. Was there something unique about them that we could have known prior to that moment of first diagnosis that tells us something about their risk of rapid progression? And then utilization insights and even applications to clinical trials, saying where do trials fit into that patient journey? Where might it be appropriate for a patient to access a new therapy that's still being tested, but that might meet a need they have? These are also ways that we can enrich the patient journey using this digital phenotyping technology. To make this a little bit more concrete, I'll go through a few different case studies. And the first of those corresponds to the diagnosis landscape. As I mentioned, this is probably the most common question that we get asked at OM1 when we're trying to use digital phenotyping to enhance understanding of the patient journey. So some of the key challenges here, I've already mentioned it several times just because it's almost a passion project of mine to help solve some of these mysteries using AI and digital technology. But this question of underdiagnosis and misdiagnosis cannot be overstated. Probably everyone on the webinar right now is familiar with a disease in their area of specialty where they could point to issues of underdiagnosis and misdiagnosis. This is prevalent across many different conditions, many different therapeutic areas. And it's not random underdiagnosis, by which I mean it's not like, 50% of people are diagnosed and the other 50% of people are still missing diagnosis, but they all look the same. A lot of times, as I had described a bit ago, those groups look different. And if we are basing our analyses, our studies, our commercial plans on unrepresentative populations because we only focus on the people that we can see, and those aren't reflective of those we can't, we're going to miss things about the real world patient experience as well as the real dynamics of who can be accessing treatment, who can be helped by treatment, and so on. So this notion, as I said, of underdiagnosis, misdiagnosis can really not be overstated in its importance. Further than that, many conditions that we care about lack clinical coding. So the easiest way to begin constructing a patient journey with respect to a condition is to say, what patients do we have in a data set who have coded information of that data set, right? There's an ICD-10 code or whatever it may be that tells us, that patient was diagnosed. Many conditions don't cooperate with that because there is no clinical code for that condition, or there's a code that's used to represent the condition we care about, but several others as well. And it's difficult to distinguish, what is meant by a provider when they use that code, whether that's really the cohort we're most interested in. Of course, there are plenty of tools in our traditional toolbox to deal with some of these issues. We have things like operational definitions where we'll say, perhaps on top of a diagnosis code, we're also interested in treatment codes that point us to one direction or another, that tell us the physician was focusing on this patient, sort of down one branch of a journey versus another. The problem with those, as anyone who's worked in implementing them in data sense, is that they're not perfect. They can be contradictory. Sometimes we'll have operational definitions that contradict one another for the same condition. They can be hard to implement in data sets, just from a technical point of view. A lot of times missing data frustrate our ability to apply operational definitions, and they're almost always incomplete. They miss patients, and I'll show an example of that in a moment. And even patients that accurately get diagnosed with a condition, even through any of these complexities, may still experience significant delays between when their symptoms start and when they reach that diagnosis. We've done a good amount of work in patient identification and journey enhancement for rare diseases. And there, the delay between symptom initiation and diagnosis can stretch for years, if not even longer than that. Collapsing that interval and trying to say, as we observe what's going on with a patient, can we say earlier, here's where this looks like it's converging for you, and therefore, it may be useful for you to consider treatment, get diagnostic confirmatory testing, whatever it may be. To collapse that interval is also a big challenge with respect to diagnosis. So here's a case study example that's really associated with this question of diagnosis, and it comes from the world of mental health, and in particular, treatment-resistant major depressive disorder. So literature will give all sorts of varying estimates about how prevalent treatment-resistant depression is. I've seen everything from 5% to 30% or even more of major depressive disorder patients are estimated to experience treatment-resistant depression. One reason that estimate is so wide is because there is no standardized coding for this condition. There are definitions, but as I'll show you, they don't give the same answer in each data set or even when placed next to each other in the same data set. And that makes it hard to understand what's going on with this reality. It's important because, treatment-resistant depression, of course, is something we want to understand if we're thinking about treating depression. We want to know how many patients are resisting treatment, why that's the case, why their depression, doesn't respond to the agents we have available. And even more importantly, what we can do for them, with new waves of therapy, right? It's very powerful to be able to say, as is the case right now in clinical development, that there are new medications coming out specifically for patients who have experienced resistance to other forms of treatment. But we won't know to match those new treatments with our treatment-resistant patients unless we have some ability to put our finger on them and say, here's who they are. So there's the key question that sort of frames this issue right now in understanding treatment-resistant depression patients' journeys, which is, we have several definitions. Do they capture the same patients? So, we could start with the regulatory definition. This is, the definition that's most commonly used by regulators when considering clinical trial endpoints and criteria for examining new treatments. That says 2 failed sequences of treatment of adequate dose and duration within the same depressive episode. There are a number of challenges with that one, but the biggest one I'll point to is this word adequate. There's no standard meaning to adequate that lives within this definition. That can differ from psychiatrist to psychiatrist. And so already we're seeing there's some potential for variation in what this means. There are several other definitions that have been constructed for TRD. I like to start by describing, what we might think of as gold standard definitions. And what we mean by that is a patient meeting either definition is objectively TRD positive. One of those we could say, well, let's go into the data set we have available. Let's look through patient histories and let's look for one of these advanced therapies. This is the gold box on the left. So for example, electroconvulsive therapy, that is not something that a patient is going to experience unless a provider has said, this patient has failed a lot of other treatments. They still need a lot of help. Let's see if this will work. So it's pretty incontrovertible that if we see that piece of evidence in a patient's journey, then at least by that point, they've reached sort of a designation as experiencing treatment resistance. Another option we have is to say, let's look at clinical narratives, right? Not structured data in our health history records, but actually the notes and other information recorded by psychiatrists. And let's look for instances where the psychiatrists attested that a patient was experiencing TRD. And you can do this. There's no lack of these things available when we get into unstructured data. And thankfully for me and for our group at OM1, we have access to tremendous richness in terms of clinical narratives to pull out exactly this sort of thing. Now, based on those 2 gold standards, we can go a little bit further. Of course, the problem with the gold standard that depends on sort of very advanced therapy is that when we see it, we know a patient has experienced treatment resistance, but we can't say anything about patients who haven't experienced these very advanced therapies, and that includes most patients. You can certainly be treatment resistant without having progressed to electroconvulsive therapy, for example. So several researchers in the paper that I've included a slight citation there too, came up with a proxy for this data-driven gold standard definition. And the proxy that they came up with was an observation of at least 3 different antidepressants or at least one antidepressant plus one supplementary antipsychotic within 1 year of the patient's history. So in other words, if you see that evidence in the journey, it is okay to label that patient as having experienced treatment-resistant depression. It's supposed to capture the gold box above it, as well as include those kind of similar to those patients. In the other gold standard form, this physician-attested definition, we used digital phenotyping to try to approximate that definition. And in particular, we said, let's look at patients, a cohort of patients who have attested TRD in their records. So in other words, there's a psychiatrist saying this patient was experiencing TRD, and then let's phenotype them. Let's say in those patients' structured records, now not relying on the unstructured data, but in their structured records, can we assemble one of those fingerprints of the sort that I showed you? That tell us something distinctive about these patients relative to other patients experiencing depression. Once we have that fingerprint isolated, let's compare people to it and say, are there people out there who look very much by their similarity like they're experiencing TRD, because ultimately they look like those that the psychiatrist described as having TRD. And just to show you an example, this is a snippet from a psychiatrist's note that very clearly states, this patient is experiencing treatment-resistant depression. So we can be quite confident in that gold standard that we can extract from the notes. The challenge is, of course, you don't always have a note on everybody, and even when you do, the physician isn't guaranteed to have written down treatment-resistant depression, even if it's true. But we at least can get a reasonable sample size to start, and then do phenotyping on to translate to the structured data. I'll give a little bit of the punchline. When we use digital phenotyping and our phenome technology here to identify these patients, we perform quite well. I won't belabor the statistics here, but those are of you who are familiar with AUC metrics, 0.87 is quite a good score. And this is, reflecting the underlying technology's ability to isolate TRD patients within an MDD pool. So the testing here was good, but I think more impactfully, we might say, how do these three patient cohorts overlap, right? Getting back to our journey question, what we want to be able to do is say, if there's something common about treatment-resistant depression, we should be able to notice what those patients' journeys have in common. And we should be able to describe that, study it, understand how to intervene earlier, and understand how those patients differ from those not experiencing treatment resistance. So if these three definitions that I described are perfect, I'll say, they should all capture the same patients, right? Because there should be treatment-resistant depression patients out there. It shouldn't matter, which definition we use if all of our definitions are equally valid. Long story short, out of about 45,000 patients that we found within our major depressive disorder data set who have been identified by one of these mechanisms, the question is, what overlap would we expect among these three different cohorts? So the answer is 1%, which shocked me perhaps as much as it shocks any of you based on your responses. Only 1% of patients that were identified here actually met all three definitions. What that is to say, in other words, the other 99% of patients were missed by at least one definition. And in particular, 9% of patients met two definitions. The remaining 90% met only one definition. That's pretty dramatic, because if we go back to how we came up with these definitions, they all have some reason to exist, right? We all sort of have some sort of initial validity to think that they are going to capture treatment-resistant depression. But in fact, patients get missed. They fall through like Swiss cheese from these different definitions if you try to capture the same patient using more than one of them. 10% of patients within this big group met only the phenome definition. And this is really important because remember, that definition is the one that is approximating what psychiatrists consider to be treatment-resistant depression. So if 1 out of 10 patients who are identified here are only getting caught by this digital phenotyping model and they're being missed by the more traditional ways of looking at this patient population, that means there's a lot of patients out there who the psychiatrists would agree are experiencing treatment resistance and are in need of advanced therapy, but who we won't study or we won't build into our planning if we don't have this sort of way of accessing those patients, because they're missed by the other ways that we'd have to define those patients. So what are the implications of all of this? First of all, it's good to see that we have a reliable process for taking patients that physicians kind of label or say, I know it when I see it, and here's what it looks like, and translating that to structured data, translating that to much larger data sets to say, based on one of these phenotypic patterns, here's the patients who look like those that the physicians designate. The second point, patients identified using the technology don't always overlap with other definitions. And as I was just saying, that's really important. If we're building out a plan of how to reach patients with a new treatment modality, and we're not aware that these patients are out there and being missed, we won't plan for them. And that's really bad for them. And it's bad for us because we have something that could be helpful to them. Without this kind of insight, we are at risk of the same kinds of bias I was describing up front as well. It is true that when patients kind of fall off the radar, if enough of them fall off the radar and look similar to one another and different from others, we're missing a huge demographic group. And in fact, when I've gone and looked at some of the individual cases in this previous case study with some clinicians, they've said, yeah, this reminds me of people in my practice who are actually the most severely treatment resistant patients. They've been so burnt out on things that they've tried and that haven't worked that they haven't tried anything recently. And for that reason, they're not going to meet a filter that depends on activity, for example, within the past year. These are not super arcane or non-obvious conclusions, but it's really important to emphasize that without this technology, we can't see these people. And they're important to see because they're able to be helped. So that sort of concludes my story about diagnosis. But as you can tell, I get quite excited about this because there's so much good to be done with it. In our second case study, I want to move from thinking about a sort of diagnostic kind of patient identification notion into something more closely associated with the effects of treatment. So here, the challenges that we see are often associated with some of the contextual information, some of the why around what happens across the patient journey as we look at treatments. So it is common, of course, in patient journeys now to observe treatment use, to observe initiation, discontinuation, and things like this. And we're always trying to understand more about those events, right? We're trying to understand the why. And we do the best we can using sort of the longitudinal data that we have, but those methods are often just not able to get at some of the factors associated with these decisions. And without that contextualizing information, two patients can look the same, even though they may have actually experienced dramatically different journeys. Again, like the example I had before, if you have one patient who discontinues because of a lack of insurance coverage of medication versus another who discontinues because of side effects, the discontinuation may happen on the same day, may look undifferentiated between the two. But in fact, those are two distinct journeys. And we need to be able to contextualize those patients' journeys to understand that difference. So what can we do? Well, one of the first things we can do, again, drawing on the strength of the unstructured data that we have access to, like I was describing in the previous case study, is say, can we isolate patient groups here based on things we can read in their narrative, and then go observe them more closely in the structured data. So for example, here's 3 instances, again, for mental health, where we have a patient who had to stop Lexapro because of diarrhea. Fluoxetine was associated with severe insomnia. Sertraline caused stomach cramps. These are not unknown side effects associated with use of these medications. But here, we sort of have the smoking gun, particularly in that first snippet, right? There's evidence of the discontinuation, stop Lexapro. And then there's the causal link that says because of a side effect, right? That is a very valuable piece of information that can strengthen our understanding of the patient's journey and can kind of buttress that causal connection between maybe a report of the side effect and the instance of discontinuation of the medication. Another thing we can do with respect to digital phenotyping is use it to understand treatment-based phenotypes. And the notion here is to say, as we're trying to consider patient journeys, we can begin observing whether patients are sort of grouping into different areas based on who responds to what. And this is particularly useful when we're thinking about a complex therapeutic area with lots of different options that patients and providers have access to, where there's not always the most simple, let's say, treatment choice algorithm, right? This is not the kind of case where it's tried treatment A. If treatment A fails, try treatment B. There might be a range of treatments available, and providers, may individually have their favorites. They may, have some literature-based information to kind of nudge patients in one direction or another. But we can take a more robust data-driven view of this and say, are there phenotypes associated with patient response to treatment A versus treatment B? And in this case study, I'll just show some examples of, advanced too fast there. We were analyzing two treatments in the area of vascular disease that I've just called target treatment and non-target treatment. And we were looking at patients' downstream risk of amputation. That was the thing that we were trying to focus on. Does the target treatment or the non-target treatment work better as defined by a reduced risk of amputation for patient A versus B versus C versus D? That's what we were trying to phenotype. And just to orient folks, you can kind of see from the coloring that the most extreme phenotypes, if I can call them that, are the ones that point most directly towards a patient journey in one direction or another. So over here in the dark red, patients who have a phenotypic profile, and I'll show you some examples of what these profiles look like in a second, but patients who would have this profile are likely to do worse with a target treatment and to do better with the non-target treatment. So for that patient, it's likely that the non-target treatment is the one that probably makes more sense for them. And similarly, at the opposite side, we have these patients who are likely to do worse with non-target, better with target, and those are probably the most natural candidates for the target treatment. And you can see some of the sort of intermediate cases. Of course, the gray ones are the ones where choice A versus choice B is a little bit indifferent, right? That journey doesn't necessarily split one way or the other. But those who are in the colored squares are sort of more likely to be helped by taking one path versus the other. The question is, can we look at a patient's journey and say, this patient kind of belongs on this path versus that path? I will say, we don't often see stuff in the dark corners of this graph. And the reason here is because if it were known, if it were so obvious that, this patient A, is going to do very well with the target and not so well with the non-target or vice versa, that would be a treatment algorithm of the sort that I had just described, right? In this case, it would be, patient who looks like this, looks like these demographic descriptors, perhaps has these lab values and so on. Definitely start them on target treatment, and then if that doesn't work, consider something alternative or vice versa, right? And this is actually how lots of treatments work. Lots of precision oncology treatments require very specific parameters, but within those parameters, those treatments work quite well for patients. Not the case in lots of different diseases, again, where it's not so obvious that there is a particular phenotype of a patient, a particular kind of patient for whom a treatment will or won't work relative to another treatment. But more interestingly, there are some of these areas where we can definitely say a patient is a little bit more likely to do better on one path versus another. And I've listed some of these things here. For example, over in this light green box, we found a phenotype of patient that was associated with atherosclerosis history, prior evidence of thrombosis, various cardiovascular issues, patients who had a phenotype defined by some of those factors, and of course, I'm not doing justice to the phenotype, they really would do better when we looked at their data with target treatment versus those who are exposed to non-target treatment. And this is informative because we can now say, okay, we have this phenotypic profile. People who look similar to that, may want to think about one treatment option over another. Their physician may want to consider that a little bit more closely. Perhaps we want to study that group and see about, how an indication is associated with that group a little bit more closely. But rather than where we started, which was a whole mass of patients all progressing along on a journey as a big clump, we can now say, okay, those who have some similarity to this descriptor on the left hand of the slide, maybe we think about them as having a separate journey. Maybe we study them, as a separate group and target them as a separate group for the therapy that's most likely to work for them. And you can see some similar identification in the other light green box. And then we also found some parallel sort of information in the light red boxes. Again, just with those 4 different boxes, we now have 4 different potential journeys that we split out of one big journey where we started. And we can say these patients, stream this way, these stream that way. Is there some validity or some utility to formalizing those notions now that we've understood them? So using digital phenotyping to cut through some of these complex journeys where some patients will try one thing, other patients will try another. There's not great standard practice in the field of what's used when. A lot of it's kind of anecdote dependent. It can be very clarifying to use this method to say which kinds of patients do better according to one of these channels. And again, the thing to emphasize here is just that the raw material from patient records, including that information we can get from the unstructured clinical narratives, is so informative when we try to, have richer understanding of patient journeys with respect to treatment. Orion is really well positioned, well designed, I think, to take advantage of that richness, and we can augment our understanding of what's going on with patients so much when we have access to that information, and then we can translate it to patients in poorer data environments as well. As I was just describing, the treatment-specific phenotyping can break patient populations into meaningful subgroups and meaningful subjourneys, right? We can clarify or crystallize what's going on with a certain kind of patient as they progress along a treatment decision path. And ultimately, this can really help us shine light and insight onto which treatments best match which patients. Final case study, to make sure that we have plenty of time for wrap up and questions, is around identifying and isolating key disease subtypes. So again, here, this is sort of related to the previous treatment associated example that I gave, but the common problem, I think that crosses a lot of therapeutic areas, and that's probably familiar to the entire audience, is that a lot of patient cohorts look uniform at index. And even if we can see that, if we peer backwards into those patients' journeys, different things were going on, we have a notion that they may be distinct. It's hard classically to connect some of those prior heterogeneities to what will be different in those patients' future outcomes. And this is all particularly true in the areas of disease that are most, messy, for lack of a better term, where we have a large disease category that does not have well-defined subphenotypes, but where we know there's all sorts of complex interaction with different conditions and different demographic characteristics. I mean, obesity is probably a great example. I'm sure some people talk about subtypes of obesity. I wouldn't say that that's a hugely widely accepted way of classifying obesity, except by BMI categorization and perhaps overlap of certain specified metabolic comorbidities. But I think we all have an intuitive notion that obesity is a tremendously complex disease, in particular in its overlap with other comorbid conditions and in terms of other factors in patients' lives that intersect and interact with their obesity. For example, some sociodemographic characteristics that patients may have can influence how obesity manifests, how it may be best treated or managed. All of that complexity is sort of, lost when we describe someone purely as a BMI number. And introducing or reintroducing richness into understanding distinct patient journeys there is what we use phenotyping to do in this sort of application. And so I'll show you an example here in metabolic disease, actually. But before I show the example, I'll just describe whenever we do one of these subtype associated journey analyses, we'll think first about the health history that the patient had prior to whatever indexing event we choose. And the indexing event can be anything. It can be, the first time patient got diagnosed, or the first time they had an abnormal lab value, or whatever we want. But that's sort of the common starting point, and we'll pay attention to what was known about those patients before that starting point. Everybody sort of is at the same point on the starting line when we've defined this index, but after that is when we start seeing some splitting into clinically distinct patient journeys, right? This is where, one patient may progress faster than others, or one patient may, evolve a certain subclass of symptoms that others don't experience, or whatever it may be. Again, can be anything, but there's just a notion of distinctness here. And then we can even get, further down and split some of these journeys into secondary and tertiary subjourneys. And a lot of times, as I was describing in obesity, the complexity gets overwhelming here. There's so many splits and forks and distinctions that it becomes very hard to keep track of who is on which journey and who is sort of like or similar to others, in where they are on that journey. But the good thing that we do with digital phenotyping is say for patients who match this branch, how can we describe, a common phenotype that those patients share from their health history prior to where they started, prior to all this splitting happening? And once we have that phenotype set up, we can connect those points in time. We can say for new patients, before or when they get to the indexing event, we don't know which split they're going to take, but if they match that fingerprint, then they're most likely to follow that branch on the bottom, for example. And we can do this with all the different branches, right? And with this library of phenotypic fingerprints that we isolate, we can prospectively match and map patients likelihoods to take, the top one versus the middle one versus the bottom one versus some of these other ones. It provides a lot of illumination into what will happen most likely to these patients down their journey. Here's a practical example of what I'm talking about. This is some work that we've been doing over the course of a number of years. We've just had some posters come out on it, which I'm excited about, and are working on a manuscript in concert with Novo Nordisk on this, which is quite cool too. Here, the patient experience is associated with MASH, formerly known as NASH, and the indexing event is just patient's first diagnosis with MASH. And of course, the health history prior to that diagnosis is what we're using for phenotyping. Here, we've split their trajectories in quite a number of ways. We've looked at rapid progressors, right? Patients, in this case, whose liver fibrosis progresses quite quickly. We also looked at patients who progress to severe long-term outcomes in their disease, things like liver failure, and patients who, down the road, 2, 3, 5 years beyond their initial diagnosis, are going to have disproportionately high resource utilization. And we did some tertiary splitting as well, looking at, in the case of severe long-term outcomes, those who progress to liver outcomes versus severe cardiac outcomes. These are not comprehensive splits. It's not the case that all MASH patients fall into one of these journeys, and they can overlap, right? We can have a rapid disease progressor who also progresses to severe long-term outcomes. Nothing invalid about that. The point is not to say this is a complete reflection of what's going on with patients in MASH. It's to say we can be precise about subgroups we care about, and then we can phenotype those subgroups to connect them back to what was known about those patients before they split down their journey. And this is just a little screen grab from one of the posters that we have here, where in particular we isolated a phenotypic profile of patients who are much more likely to experience rapid disease progression based on what we knew about them prior to first diagnosis. And we don't have the luxury of more time in this webinar to go into the detail here, but just at a high level, this cluster of factors is both important in identifying patients, and these factors' relationship, their sort of relative positioning to each other, is what allows the underlying technology to say this patient, as opposed to these 10 or 50 or 100 others, is the one most likely to end up going down that path. Are there distinct things about that patient we can talk about, before they end up there? For example, with respect to treatment access, right? This may be a case where the patient would really benefit from early treatment to prevent disease progression because this patient's at much higher risk of rapid disease progression. That's the kind of thing we can do when we've clarified these journey insights. And so just to wrap up this section, the key notion here is that when we're thinking about distinct subtypes of patients who all progress from a common starting point, we can use digital phenotyping to link those downstream differences in outcomes to data that we knew when those patients were starting before they differentiated. And once we've defined those subphenotypes, we can prospectively track new patient populations and observe them independently, right? We can say, we think this group is going this way. Maybe there's, as I said, a case to be made for earlier treatment for them, or particularly bespoke outreach that we can do to that group. It provides us a lot of ability to, again, put our finger on that group of patients and say they're different from everybody else. Let's approach them in the way that makes most sense for what they need, as opposed to being sort of washed away into the larger group. So we're almost out of time, but I wanted to mention two other things with respect to the application of digital phenotyping to patient journey analyses. The first is, how do we deliver all of this information, right? I've described lots of insights that can be added to our understanding of journeys using digital phenotyping, but how do we actually consume those insights? One way is to simply report on them, right? In a sense, that's what I've been doing today. I've been describing the results of analysis we've done to enhance patient journeys using digital phenotyping. But this information can also be consumed in a very comfortable way for researchers. If folks are used to looking at data themselves and constructing their own journeys using those data, it is quite easy to simply add phenotype-derived information to those data sets and use or not use that information wherever it's useful. So, in this example on the top, we might say, here's a patient who's a male, born in 1972. They belong to a group of patients we've identified as having psoriasis. So for example, they've met certain inclusion criteria to say, we're studying psoriasis journeys. this patient fits. We could add to that, a couple more columns that say, within psoriasis, there's a subgroup of patients at elevated risk of progression to psoriatic arthritis. Does this patient match that phenotype, right? In this case, yes. And then we can also say, here's some confidence associated with that assessment. And then, we can study journeys with that information. We can add it, subtract it, be precise about what confidence thresholds we want to use. This is an augmentative tool for research for folks who are used to working in the data, and for those who prefer kind of the summary results, that can also be just delivered at a higher level. And so just to conclude, so we have a couple of minutes left for questions. Patient journey analyses as currently conducted do have significant gaps, and hopefully, I've highlighted a number of areas in our understanding of subtypes, of diagnosis, of treatment pathways, where it is very helpful to patients and to us who work in these areas to understand what's going on. Digital phenotyping can be used to fill in some of these gaps, particularly in those areas I just mentioned. And these phenotyping enhanced journey insights can be used side by side with more traditional data and analyses. The title of this talk was Practical AI, and hopefully I've demonstrated that this AI is not exotic and crazy and weird. It is very useful side by side with those ways that we already have of consuming information and constructing journeys. It doesn't need to be arcane and throwing out all of the good methods that we have already to understand what's going on with patients. And finally, through phenotyping, I believe and have experienced quite a lot in my own work and in our work at OM1, that we really can tackle some of these patient journey questions that have remained unanswered using traditional methods alone, with significant impact for us in the life sciences industry, for providers, and most importantly, for patients, because the more clarity we bring to what's going on at the individual level, the better we can match those individuals to the treatments that can help them most. And so with that, I think hopefully I've left a couple minutes for questions and happy to take them.
Great. Thanks, Joe. That was a really great presentation. And thank you for mentioning the ebook. Okay, let's jump right into the Q&A session. We only have a few minutes left, so we'll get right started. Are there any clinical areas that you found helpful in which this approach could be, where you found this approach particularly helpful?
Yes. So there are a number that stand out as particularly useful for digital phenotyping to shine additional light on. One is in those areas of complex disease. I was giving some examples from metabolic disease, but certainly immune-mediated disease. Mental health can be quite complicated and difficult to differentiate patient paths. Those sort of large and messy disease areas are quite amenable to this kind of clarity. The other one, interestingly, kind of at the opposite end of the spectrum, can be rare disease, because in rare diseases, there's so much of this off the radar problem where patients are bumping around from misdiagnosis to misdiagnosis. Small populations, so we already have difficulty estimating prevalence and understanding what's going on. So that's another area where this can be quite fruitful.
Okay, wonderful. Thank you for breaking that down.
Mm-hmm.
Our next question, did you compare this predictive diagnosis with the consistency of diagnosis across physicians? So for example, if you present the same parent to multiple physicians, how often do they produce the same diagnosis?
Oh, that's interesting. So would we do testing to say, here's an individual sequence of data, what would physician A versus B versus C kind of diagnose consistently? We haven't done that in terms of, I would say, physician panels, but we do this in a way by saying if physician A versus physician B are looking at two patients who look quite similar, do those two physicians arrive at the same conclusions? And sometimes the answer is no. And then we have to be very careful about, which ones we're using for calibration. In most instances, I would say the physicians are reasonably consistent within a specialty area. We do see quite a lot of divergence if you say, let's compare, non-specialists with what specialists diagnose or conclude from a particular sequence of data. We've done a lot of work in dermatology in this context, for example, looking at patient journeys to diagnoses of dermatologic disease when being treated by a dermatologist versus reaching those diagnoses through other paths, through primary care, urgent care, things like that. And those can be quite different. So it's important to understand the subtlety there.
Okay, thank you. Great answer. Um, Somebody said, thank you for the insightful case studies. They would love to understand more why Phenome versus other RWD analytics or providers would be chosen.
Oh, okay. Yeah, I mean, I would say quickly, just because I know we're close on time. The thing that's different about digital phenotyping and phenome, the way we do it here, relative to others, is I think a lot of AI companies, a lot of folks using AI and machine learning tools, often focus on prediction as their first task, right? Predict who is going to end up here, predict who has this disease, that sort of thing, and that's fine. It's a useful thing to do. What it doesn't focus on is what is distinct about those patients in a way that we can go into and describe and explain and work through with clinicians. And also, once we have a phenotype, we can ask different questions of the same patient's data, right? We have, a set of data that give an individual patient's phenotype, just like our genetic records all give us our own genetic profile. And once we understand that about an individual, we can say, does this patient have this risk? Do they have that risk? Are they likely to correspond to this treatment? That sort of ability to ask multiple questions of the same set of data is a little bit different from how others do this, and is quite fruitful, again, in journey analyses where we're not usually restricted to just one question. We want to ask many.
Okay, clear and concise answer. Thank you. We are at the top of the hour, but if you have one more minute, I think let's squeeze in a last question.
Sure, let's do it.
All right. Can digital phenotypes be applied to different data sets? For example, to compare how many patients are found in one data set versus another?
Yes, absolutely. So this is one of the really nice things with phenotypes derived from rich data, like we're lucky enough to have here at OM1, because there are many paths into the phenotype. You can take that phenotype and apply it in other data environments, and then do exactly what the question is asking. Say, what rate of patients do we observe in one data set versus another? Are they consistent? Is that evidence of something meaningful about the patient population? So yeah, translation to different data sets is important here.
Okay. Thank you so much. It was such a pleasure to have you with us today and for answering all those questions. And yeah, I hope you enjoyed yourself.
I did. Thank you to you and to the audience for your attention.
Fantastic. In closing, we hope you enjoyed this HealthEconomics.com webinar sponsored by OM1, and I hope you enjoy the rest of your day.